This particular Chapter 15 video is dealing with not just the metabolism in general, but more specifically the recurring motifs. And of these recurring motifs, this is going to focus on the portion that is related to activated carriers. Key reactions and regulations will be in another video. So in terms of activated carriers, this has to do with the phosphoryl transfer potential of molecules. Okay. We have seen that ATP is an activated carrier of phosphoryl groups. And we saw that because that phosphoryl transfer is an exergonic process. And we can couple that with other endergonic processes to make them more favorable. But the idea of activated carriers is not unique to that phosphoryl transfer group. There are other things that we can have serving as activated carriers. So here we see some different types of activated carriers. We have activated carriers of electrons, so we're carrying electrons, and those electrons are carried so that we can complete fuel oxidation. We have activated carriers of these electrons that allow us to undergo reductive biosynthesis. So in one case you are breaking down, and in the other case you are building up or making something. And then we have activated carriers of not electrons but two carbon fragments. So we're going to look at each of these individually. So activated carriers for fuel oxidation. There are a couple of activated carriers that we focus on. The two more common electron carriers are going to be this NAD plus and FAD. These are going to be the two most common substances that are going to take the electrons and carry them, and eventually the goal is to get these electrons to oxygen. Oxygen's our final electron acceptor as we're going through the metabolic process. But the electrons that are coming from the different molecules that we have that we're taking in and breaking down don't directly go to O2. There's not a direct transfer. So these activated carriers help take the electrons and then pass them along later as needed. So NAD plus and FAD are these carriers. So we see here in the case of the NAD plus, what happens is you have some molecule, and that NAD plus is going to pick up electrons. It picks up two electrons and a proton, and it becomes NADH. So two electrons and one proton are picked up by the NAD plus to become NADH. And then we have over here the FAD, and the FAD is going to become FADH2, and in order to do so, it picks up two electrons and two protons. Okay, so these are the two activated carriers of electrons that we're looking at in terms of fuel oxidation. So here's the structure of the NAD+, the nicotinamide adenine dinucleotide. And in this structure, the place where we're having the electrons being picked up or being transferred is going to be in this reactive site here. The NAD+, this is one of the major electron carrier molecules. So that ring portion up there, this ring portion, is going to serve as the reactive portion, and it's actually going to be, this portion is going to be synthesized from niacin. Okay. Okay, which is a vitamin. The nicotinamide ring of this NAD+ is going to accept a hydrogen ion and two electrons, and when it does so, it's going to become that NADH. So the NAD+ is going to serve as an electron acceptor in many reactions. Now we also have, I mentioned this in lecture briefly, in addition to NAD+, We can also look at NADP+. This is another electron carrier substance. Now the difference between the NAD+, which is shown here, and the NADP+, is going to be that in this structure here, for the NADP+, this R group is going to be a phosphate group. Whereas when we're looking at NAD, that R group is going to be a hydrogen. So for NADP+, it's a phosphate. For the NAD+, it's a hydrogen at that position. That's the only difference, but they're both electron carriers. Now another one of these electron carriers is the flavin adenine dinucleotide, FAD. So this is another major electron carrier. The FAD is the oxidized form of FADH2, which is the reduced form. What happens here is this substance gains electrons to become FADH2. And this portion that's the reactive site is up here, this ring structure. And this ring structure is referred to as an isoalloxazine ring. And it is actually derived from riboflavin, which is another vitamin. So that's the reactive portion. And I'm highlighting this because we're going to talk about vitamins at some point, and just to point out that where their significance lies. This is going to be derived from that riboflavin. Now here, FAD can accept 2 electrons and take up 2 protons in the transfer process. So 2 electron carriers, NAD plus and FAD. Now here what we're looking at is just the 2 different forms of that FAD and FADH. So you see the oxidized form, and this form picks up 2 hydrogen and 2 electrons to become the reduced form, which is the FADH2. So these are our activated carriers of electrons for fuel oxidation. Well, now we can talk about activated carriers for reductive biosynthesis. When dealing with biosynthesis, the precursors are more oxidized than the products, so we actually need high potential electrons. These high potential electrons provide reducing power that is needed. We also are going to have to use some ATP when we're synthesizing these things. So here what we're looking at, this just illustrates one example, and in this example, if you look at the molecules that are involved, what we're doing here is this represents the biosynthesis of fatty acids. So in this case, we have a keto group that's reduced to a methylene group, and that happens, we're showing it here in one step. So this group is being reduced to that group. It's shown here in one step, but the reality is this occurs through a several-step process. Throughout this process, in order to have this keto group being reduced to a methylene group, there will be 4 electrons and 4 protons that are involved. So we have electrons are being carried here. Now we saw that FAD and NAD plus are the electron carriers that we're going to see in fuel oxidation. And we talked about how we had NAD plus, but we also have NADP plus. The NADP plus, this electron donor is going to be most widely used in biosynthesis. Okay, so NADPH is what's most widely used in biosynthesis, and it's going to be that electron donor. And again, the reduced form is NADP+. The NADPH and the NADH will carry the electrons in the same way. So this carries electrons in the same way is NADH. But the NADPH is used almost exclusively for this reductive biosynthesis. Whereas the NAD is NAD and NADH, those are going to be used in oxidation. NADH, again, oxidation primary use is generation of ATP. In the biosynthesis process, remember that we are carrying these electrons, but we're also using ATP. NADPH is used in the biosynthesis. And that extra, the NADPH version, this has that extra phosphoryl group, like we talked about when we were looking at the structure. And that extra phosphoryl group is a tag that helps us distinguish between the high potential electrons that are used in anabolic pathways versus those that are used in catabolic pathways. So here where it's biosynthesis, we're building up. So building up, it's the anabolic processes. So we're using that NADPH. In the catabolic processes, the breaking down, that fuel oxidation, we're using the NADH. And now moving to the next activated carrier, we have two carbon fragments that are activated carriers. Coenzyme A is a carrier of acyl groups. Okay, so this coenzyme A is a carrier of acyl groups. Acyl groups are derived from a vitamin, and that vitamin that they are derived from is pantothenate. The acyl groups are going to be significant for both catabolic and anabolic processes. So it's not just, we're not just talking about fuel oxidation or reductive biosynthesis. Both of these types of processes, the building up and the breaking down, are going to involve acyl groups. On this particular structure, the reactive site that we're looking at is the sulfhydryl group that's present, and it's kind of highlighted here. When the acyl group is acetyl, this is acetyl-CoA. And acetyl-CoA is a substance that we are going to see used in other processes. One of the goals when we have the breakdown process of glucose undergoing glycolysis, and we're making pyruvate, and then acetyl-CoA is one of the fates of pyruvate. So this is going to be important. Well, the hydrolysis of the acetyl-CoA has a large negative free energy value, and that acetyl group, that's going to go into the Krebs cycle to be broken down. But the CoA, that coenzyme A, is carrying that acetyl group so that it can go into the cycle. So the activated carrier of the carbon fragment is the coenzyme A, which is carrying the acetyl group, and the acetyl group is what's going to be broken down, and CoA is going to be regenerated. So this is just looking at the energy that's associated with this process and illustrating the difference in stability related to using oxygen esters versus thioesters. So when we're looking at acetyl-CoA, it is a thioester. That's what we're looking at. And so the reason that we are going to use thioesters is partially based on how the resonance structures are going to stabilize these things. So the oxygen structures are more stabilized, and the energy is going to be different there. So the ability to transfer substances changes because the thioester portion, because this is going to be less stable. It's going to be better at, or it's going to be used as, a transfer group. And here all we're looking at is the structure of that coenzyme A. So coenzyme A in general is outlined here. this is the coenzyme, and then this reactive group is what would be attached to that acyl portion or the acetyl portion. So we can look at the activated carriers overview. There are two common characteristics of activated carriers. The carriers are kinetically stable in the absence of specific catalysts, and the metabolism of the activated groups is accomplished with a small number of carriers. So in terms of reacting in the absence of a catalyst, NADH, NADPH, and FADH2, these all react slowly with oxygen when we don't have a catalyst present. ATP and acetyl-CoA are also going to be hydrolyzed slowly in the absence of a catalyst. The fact that we have kinetically stable substances, so they react slowly in the absence of a catalyst, right? So they need to be kinetically stable, again, reacting slowly in the absence of a catalyst. Because that's the case, it allows enzymes to control the flow of electrons. And because we can control the flow of electrons, the reducing power of the substances are going to be controlled as well. So this kinetic stability, that kinetic stability is due to the fact that these substances react slowly when we don't have a catalyst present. If they react slowly, it means they're stable. And because of that, enzymes can control enzymes control flow of electrons, and enzymes are going to control reducing power. So in terms of the activated groups and their carriers, we can look at Table 15.2 to see some of the activated carriers that are involved in metabolism. And the point of this is to show that despite the fact that we have a variety of different processes that occur, the group carriers are going to be small in number. So there's a small set of molecules that serve as carriers that will carry out a wide range of tasks. And again, we've looked at ATP and the fact that it has that phosphoryl transfer potential, so it makes it a phosphoryl group carrier. We've talked about the NADH, the NADPH, the FADH, and how those carry electrons. We haven't talked about it, but we also have this FMNH2 that serves as an electron carrier. We talked about the coenzyme A and how that was going to be that 2-carbon activated carrier, which is that acyl portion. And again, you can see a couple of others that as we explore further biochemistry we'll encounter. Not likely this semester, but if you are taking second semester, they'll pop up somewhere. And we can see that these substances do have vitamin precursors so these vitamins are going to be important in allowing for the formation of these carriers. So this is just highlighting nearly all of the activated carriers serving as coenzymes are derived from vitamins. We know that vitamins are organic molecules. So here we're just showing some of the structures of the vitamins that were in the table. These are just some of the vitamins that serve as coenzymes. But remember, we said that the riboflavin was part of that structure of the FADH, and so you can see that similarity there in that structure. So just to pictorially have a visual of the structures of these substances, but these are going to play a role in the formation of activated carriers. Now, there are some vitamins that do not serve as coenzymes. So just to point out here, we saw that there are vitamins that become part of these coenzymes or part of these activated carriers. There are some that are not involved in this capacity. This table is just showing those vitamins which are not serving in that activated carrier capacity, and what the vitamins are, how they function, and what the deficiency is that results from it. So a lack of vitamin A plays a role in vision growth and reproduction. If you have a lack of vitamin A, that's what leads to night blindness, some cornea damage, and then some issues with the respiratory and gastrointestinal tract. Deficiency in vitamin C. Vitamin C is an antioxidant, and that leads to scurvy. So just some examples of other vitamins. And lastly, these are just the structures that accompany those non-coenzyme vitamins, so the non-activated carrier substances, just so you can see what those structures look like. So again, the take home from this portion of the lecture is the idea of having activated carriers, and the activated carriers that we have activated carriers for fuel oxidation, activated carriers for reductive biosynthesis, and activated carriers that will carry carbon fragments, and all of these are important processes in metabolism. These activated carriers, we've seen their structures and talked about what goes on there, but oftentimes these are going to be derived from vitamins, and so we looked at the types of vitamins and what processes they're involved in. But not all vitamins are used in order to make these activated carriers, and there are other vitamins that we need, such as those shown here, that are not part of this process. They're not coenzymes, but yet they're equally as important because deficiencies will result in significant issues for us.